Yeah. Okay. Welcome back, everyone. To start our next session, it's my pleasure to welcome the chairman of the session, Dr. Miriam Fuchs, Global Therapeutic Area Lead, Cell and Gene Therapies, Oncology, Regulatory Affairs at Novartis. Dr. Miriam, can you hear
Yes.
me?
Yeah, I hear you well. Do you hear me?
Yes, it's a great pleasure to have you here with us today. The mic
Thank
to
you
you.
so much. Thank you. I would like to thank the scientific and organizing committee of this excellent medical biotech summit for inviting to the session today to evaluate aspects of the innovative field of stem cell and cell and gene therapies and to share our experience and learnings. I would also like to welcome the esteemed speakers of this session. The vision that cell and gene therapies can deliver unprecedented and transformative clinical outcomes for patients that may enable them to live disease-free for years or potentially be cured has been realized with the first gene and cell therapies approved over the last years. However, these first therapies are merely the beginning in a field that continues to undergo a rapid and dynamic evolution with an important promise for patients globally for improved and novel therapeutic treatment options. The session today will allow us to hear some insights from experts deeply involved in this exciting field of advanced therapies. I would like to introduce Dr. Badar Lamari as our first speaker to share lessons from B-cell malignancies with chimeric antigen receptor T cells. Dr. Badar obtained his MBBS from King Khalid University in 2005 and finished his residency in internal medicine from King Abdulaziz Medical City in Riyadh and obtained the Saudi board in internal medicine in 2010. He then carried on to complete his clinical fellowship in hematology oncology in 2016, as well as a fellowship in bone marrow transplant from the Washington University of Medicine in St. Louis. He also obtained a Master of Science in Clinical Investigation on the Translational Medicine track from the same institution in 2017. Currently, Dr. Badar is a consultant in stem cell transplantation and cellular therapy division at the Department of Oncology, Ministry of National Guard Health Affairs, Riyadh. And he has multiple publications on his work on novel therapeutic targets for graft versus host disease in experimental murine graft versus host disease models. And his current research work focuses on the development of cellular therapy for cancer management. I would like to welcome Dr. Badar. Thank you so much.
Well, thank you so much, Dr. Miriam, for the introduction. I would like to thank the organizing committee for inviting me for the talk today. Today, I'll talk about the chimeric antigen receptor T-cell therapy and B-cell malignancies. And probably there will be some overlap between my talk and the talk of Dr. Miriam. But I will focus mainly in the clinical aspect of this newly innovative therapy. So let's start first by understanding what is the T-cell response to cancer cells, starting from the antigen presentation of the cancer-related peptide, and then followed by the appropriate recognition of the T-cells. And this recognition of T-cells will lead to an appropriate killing and eradication of these cancer cells. However, cancer cells can go through a multi-step journey toward the ability to escape the immune system. And this is called the cancer immunoediting process that end up with escape from the immune system. And this mainly happen through multiple mechanisms, including the effect of immunosuppressive microenvironment, by the effect of other cellular components, and also by the cancer cell itself ability to upregulate certain molecules that enable them to escape the immune system. So how to help the T cells to tackle this battle against the cancer? There are multiple ways and methodology to deal with this, and the known one is the vaccine effect. And this can enhance the antigen presentation to the T cells. But most importantly, and the one that really showed an excellent effect, is the enhancing of killing through the introduction of the anti-BDL1, BD1 checkpoint receptors. However, also cellular therapy plays a major role here. I'm talking about the enhancement of the T cell recognition of the cancer cells through what we call it the chimeric antigen receptors T cells, or the CAR T cells. So what is CAR T cells? CAR T cells basically having the specific receptors that has the ability to hijack the T cells and make them recognizing the tumor antigen directly without the need for HLA dependency. It's completely does not rely on the MHC restrictions. There are multiple generations. The first generation of the CAR construct composed of the short variable chain of the antibodies that can target certain antigens. However, did not go through because the lack of the co-stimulatory molecules that was started from the next generation of the CAR construct. And there is now currently a new generation of the third generation and fourth generation of the CAR construct. But the most successful one that is clinically applicable now is the second generation. The journey started a long time ago, like 3 or 4 decades ago, when the first generation CAR construct was generated. However, this did not have appropriate clinical effect, mainly, as I said, because of the lack of the co-stimulatory molecule that has the ability to enhance the T cell proliferation and enhance the killing effect of the T cell and the persistence of the T cell in vivo. The first, second generation CAR was generated around 2003. 10 years later, the first clinical application of this CAR construct was first reported in 2009 in patient with CLL, followed by a patient with a pediatric patient with ALL. And then in 2017, we have the first report of the pivotal study that led to the first approval of the CAR T cell, which is the CD19 CAR T cell. And since then, multiple approval happened up until recently this year. We have the last recent approval of the CAR T cell for multiple myeloma. So how is this generated? It's a complex process, and first by do the leukapheresis and obtaining the apheresis product from the patient. Then this cell product will be shipped to the manufacturing facility where it will undergo multiple process of manufacturing, starting by T cell isolation, then activation, then transfixion with the CAR construct that either using the lentiviral or retroviral that carry the CAR construct that can be safely expressed on the T cells, followed by T cell expansion, and then shipped back to the clinical institution where the CAR T cell will be infused after lymphodepleting chemotherapy. One of the first and the best studied receptors is the CD19 antigen, where this showed a successful story and led to multiple approval of clinical products using actually a CD19 as a target for the CAR T cells. And why CD19? Because it's kind of lineage restricted antigen that is expressed nicely on multiple B-cell malignancies, including ALL and B-cells non-Hodgkin lymphoma. Talking about what is the outcome pre the CAR T cell era, the ALL patient has a dismal outcome after the first relapse. This cohort of 609 patients showed that the overall year overall survival is almost 7%. With many of the experts thought that a relapse refractory ALL patient is almost an incurable disease. The same story is for the refractory diffuse large B-cell lymphoma. Well, for this cohort showed that the median overall survival for those who are relapse refractory in the rituximab era is having the overall survival only of 6 months. What is the status of B-cell malignancies in Saudi Arabia? According to the Saudi Cancer Registry in 2016, non-Hodgkin lymphoma ranked among the second most cancer in adult male and the fifth in female. In 2016, there has been a total of 82 children and 114 adults diagnosed with B-ALL, diffuse large B-cell lymphoma diagnosed in 474 adults. Recently, my colleague Dr. Musab Damraj published the data from the experience from the National Guard in Riyadh of the outcome of 85 adolescent and adult ALL patients in CR1. As you can see here in the curve, the event-free survival for those who went into transplant is around 70%, where those who did not went into transplant around 40%. Still, you can see there is a good proportion of patients who relapse even post-allogenic transplant, and usually those patients have a dismal outcome without an appropriate line of therapy for them. Currently, the CAR T-cell approved product is mainly CD19 product, CD19 targeted products. 3 of them are approved for diffuse large B-cell lymphoma, and 1 for ALL, and 1 for mantle cell lymphoma. And also more recently, anti-BCMA was approved for relapse refractory multiple myeloma. We'll talk now about the result from the pivotal trials that led to the approval of those products, starting by the ELENA trial for the Taci-cell for children and young adults with relapse refractory B-ALL. Around 92 patients enrolled into this open-label phase 2, single-arm phase 2 study with patients with relapse refractory B-cell ALL. They underwent lymphodepletion with fludarabine and cyclophosphamide. And the overall overall response rate at 3 months was 81%, which is impressive for such a patient population who are relapse refractory even after allogenic stem cell transplant. The overall survival at 12 months was 76%, and event-free survival of 50%. The next study is the ZUMA-1 for axi-cell in refractory large B-cell lymphoma. Again, this is CD19 CAR T-cell with a CD28 as a co-stimulatory molecule. The patient population were patients with a large B-cell lymphoma, including DLBCL, primary mediastinal, and transformed follicular lymphoma. Those usually a patient with refractory to at least 2 lines of therapy, and also including some patients who relapse post-autologous transplant. 111 patients enrolled in this study with the primary outcome overall response. As you can see here, the overall response rate was 82% and CR rate was 58%. With a median follow-up of 27 months, the median OS was not reached, and the median PFS of 5.9 months. The second study that led to the approval of the Taci-cell for relapse refractory diffuse large B-cell lymphoma is the Juliet study. Kind of similar in inclusion criteria for the axi-cell. And you can see here the overall response rate of 52% and CR rate of 40%, with an estimated 12-month rate of relapse-free survival of 65%. And among those who went into CR is impressive 79%. The most recently approved CAR T cell for diffuse large B-cell lymphoma or large B-cell lymphoma is the Lasocell, which has also the 41pp, which is similar to the Tacicell molecule. However, in this product, they infuse the CAR T in a 1-to-1 ratio of CD4 versus CD8 CAR T. The inclusion criteria here is unique by allowing a patient with secondary CNS disease, and also allow patient who had previous exposure to CD19 CAR. As you can see here, the overall response rate of 73% with a CR rate of 53%, and median overall survival of around 21 days, and median PFS of 6.8 months, which is similar to the previous two products. So this is really good data for those patients who had relapse refractory disease, who usually at this stage of their disease, they go into experimental treatment or palliative care. However, this kind of treatment led to durable remission, which is something we haven't seen for a while for such refractory disease. However, this disease usually associated with significant toxicity. The two major unique toxicity for this disease is the cytokine release syndrome and neurotoxicity. Those toxicity has no understandable yet mechanism. However, there are multi-research effort to understand better this kind of unique toxicity for the CAR T cell. Luckily, there is multiple effort from multiple cellular therapy society that established guidelines how to manage this kind of toxicity and how to mitigate this kind of toxicity. And as you can see here, this slide summarizes the rate of such toxicity in the three trials: the ZUMA-1, Juliet, and the Transcend trial. In the upper panel, you can see the CRS rate, and the lower panel, you can see the neurological event rate. The grade 1 is almost about 81%, grade 1 to 2 in ZUMA trial, while in Juliet around 40%, which is similar to a study that tested the Lasocell. And neurological event, you can see the Axicell might have a higher grade 3 neurological event when compared with the Juliet and Transcend study. As I said, this algorithm summarizes how this risk of toxicity can be mitigated and how to interfere to manage this kind of toxicity by using steroid and monoclonal antibody against IL-6. Long-term toxicity, including B-cell aplasia, which can be seen up to 15% in adults with relapsed refractory B-cell lymphoma and up to 43% in pediatric patients with ALL. Cytopenia, which is also one of the common toxicity that can be seen. Rarely you see sometimes, can see some form of aplasia post CAR T cell. Infection also as a consequences of this therapy also can be one of the major side effects of this treatment. We'll talk now about the barrier to application of CAR T cell in the real world and barrier to clinical application of CAR T cell therapy. So there are some biological challenges and manufacturing challenges. The biological challenges including the antigen escape, which is one of the major mechanism for relapse of the disease post CAR T cell. There have been many patients who relapse post CAR T cell showing CD19 negative relapse, and there is multiple mechanism for how the cancer cell can evade the CAR T cell and develop CD19 negative tumor cells. Also, the on-target and off-target toxicity, this is one of the challenges for this kind of therapy. And also that CAR T cell persistence, in vivo persistence of the CAR T cell, also one of the things that cause an obstacle, and the suppressive tumor microenvironment. This slide will kind of summarize the current research effort to develop a better CAR T therapy, CAR T cell therapy that can overcome those barriers. For example, exhaustion can be overcome by the incorporation of BD1 inhibitor, and also in vivo persistence can be improved by improving on the co-stimulatory molecule and the secretion of the appropriate cytokines that lead to better persistence of those CAR T cell in vivo. Multi-target effect also we've seeing now dual and even triple CAR T therapy that target more than one antigen simultaneously, and also ways to reduce toxicity in form of safety switch and so on. In terms of manufacturing challenges, one of the things that associated with CAR T cell in general is the high cost. It's the costly treatment that can start from $370,000 US dollar for a single infusion for one therapy, and this is only the price tag for the product alone, without the associated cost of pre and post infusion, and also the limited availability of this product. It needs to be manufactured in well-established, centralized manufacturing facility that has the ability to scale up the production of this kind of therapy to to meet the clinical need and demand, which is mainly located in the US and in Europe. The other thing is manufacturing time delays. Again, it's due to mainly the centralized production of this CAR cells. Example of manufacturing delay in the ELINA study for relapse refractory ALL, there has been around 18% of patients who had the apheresis product collected did not receive the CAR T cell for multiple reasons, including poor cell growth, or death of the patient while from the disease progression while waiting for the product, or the development of interval adverse event that make the performance status of the patient to be impaired and unable to receive the product. The median time for cell infusion was around 45 days, ranging between 30 to 100, up to 105 days. This slide will show you how complex it is to manufacture such a therapy, and then T cell expansion followed by the cryopreservation of the product, then to be shipped to the treating center and to be infused to the patient. However, there has been a recent interest in the multiple academic institutions around the world for using a machine that can do the automated manufacturing of the T cell, where you can manufacture the T cell in the closed system, one single machine that can do all the process of the manufacturing, including the T cell separation, activation, transfection, expansion, then harvesting, then to be ready for cryopreservation. And there has been multiple reports from different academic institutions that report their outcome. This one just an example for one of the published abstracts in the ASH, where using CD19 construct in 12 patients, and the CR rate was 67% with overall response rate of 75%. Two patients have died because of death, including progressive disease or from the CRS. And in the right panel, you can see the persistence of the infused CAR T. At KMARC, we have been working collaboratively with the clinical part and research team in the KMARC to incorporate this kind of modality and adapt this type of therapy for our patient. We form two different committees, working groups from the clinical side and from the laboratory side to work together and collaborate to make this type of therapy available for our patient with the help from the clinical trial unit at the KMARC. Right now we have the IRB approval and we are in a process to get the FDA approval for this project. This will be my last slide. You can see here from the slide multiple clinical trial is currently active and recruiting patient around the world. Probably the highest rate is located in China. Thank you very much, and I'll be happy to get any questions. Thank you.
Thank you. Are there immediate questions? Thank you so much for this excellent overview and introduction to CAR therapies, and thank you so much for sharing your local activities to access for patients to CAR therapies with the local clinical trial, which I understand is just receiving approval. Our next speaker is Dr. Joseph Fraietta, and he is coming from one of the key centers of pioneering CAR development, the University of Pennsylvania. Dr. Fraietta has completed his training as a postdoctoral fellow in Carl Schulz's lab at the University of Pennsylvania in Philadelphia, where he conducted research on the mechanisms underlying the persistence of CAR T therapy in patients with CLL. In 2016, Dr. Fraietta assumed the directorship of the Product Development Laboratory at the Center of Advanced Cellular Therapeutics in the Perlman Center of Advanced Medicine. And during this time, he developed novel approaches for the treatment of cancer through genetic modification of T cells that contributed to the initiation of multiple clinical studies. He is currently the director of solid tumors immunotherapy labs at Penn, where his research is focused on enhancing the specificity and efficacy of genetically redirected T cells. And he is currently investigating the role of epigenetics in the effectiveness of T cell responses. He has made several important contributions to the field of immunotherapy, and his high impact findings have been published in high profile journals like Nature Science, Nature Medicines, and he has been the recipient of numerous awards. I'm very pleased to welcome Dr. Fraietta to share with us his insights on genetically enhanced T cells in designing, engineering, and building cancer immunotherapies.
So thank you very much, Miriam. It's a pleasure and truly an honor to be here with all of you today to give you sort of a snapshot of our initiatives at the University of Pennsylvania to both develop and further improve our cellular immunotherapy platforms based on CAR T cells for the treatment of advanced cancers. And the primary focus today will be on the hematological malignancies, but maybe considering the previous talk and Miriam's later talk, I'll focus on solid tumors a bit more. So since we've heard in the first lovely talk quite a bit about the successes of CAR T cell therapy, particularly in advanced B cell malignancies, perhaps I'll jump right into talking about the challenges. That is to say that despite these successes, particularly in the liquid tumor space, we still have several barriers to address. And some of the unmet needs in CAR T cell therapy are listed here, namely poor T cell expansion and persistence, as we heard about, T cell dysfunction, tumor escape, perhaps due to antigen loss or down modulation, and then of course getting this therapy to work well in solid tumor indications without toxicity has been a goal of the field and is still an ongoing goal of the field. So just to focus in on this a little bit more in the vein of improving clinical outcome, the importance of T-cell intrinsic fitness in the setting of CAR T-cell therapy, I think, is best exemplified when you compare the efficacy of the same CD19 CAR construct that we developed in collaboration with our partners at Novartis in two different advanced leukemias. And so one is acute lymphoblastic leukemia and the other is chronic lymphocytic leukemia. And so we and others, as you heard about, have achieved really high complete remission rates of over 80% to 90% in some trials in the setting of pediatric ALL with the anti-CD19 CAR developed at the University of Pennsylvania. But we've only observed a complete response rate of about 25 to 26% in chronic lymphocytic leukemia. And so the potential reasons for disparities in CAR T-cell therapeutic efficacy between these indications are listed up here, and they can be attributed to innate humoral and cellular immune deficiencies, including in these intrinsic T-cell defects that I'm referring to, that are characteristic of certain cancers like chronic lymphocytic leukemia, and that often worsen with disease progression. So as you can imagine, the above factors, either alone or in combination, may influence CAR T-cell potency and effectiveness. And clinical outcome, of course, as I'll elaborate on, is tightly tied to this intrinsic T-cell fitness. So we've conducted several careful investigations of responders and non-responders in CD19 CAR T-cell therapy of advanced leukemia, and I'll talk about chronic lymphocytic leukemia here. And our results indicated that clinical response to CAR T-cell therapy in chronic lymphocytic leukemia really depends on CAR T-cell proliferation and in vivo persistence. But in some cases, we can't expand enough CAR T-cells to meet the dose, particularly for heavily pretreated subjects, so we can't really even get out of the gate in this situation. And then in other instances, even if we get adequate ex vivo expansion and we can manufacture the products, patients who don't respond following infusion really demonstrate limited in vivo proliferation and essentially no persistence of those products. And so, we did a deep dive into this, and our extensive biomarker studies really revealed that even at the time of T-cell manufacturing, CAR T-cells from complete responders, when you look by RNA sequencing, as you can see here, are enriched in transcriptomic programs of early memory T-cell differentiation and self-renewal capacity, sort of what you would expect, right, from many preclinical studies. But if you look at the non-responder patient CAR T-cells, they exhibit signatures associated with T-cell exhaustion. But the good news here is, I think, and as we treat many more patients and do more of these retrospective analyses, we are actively identifying pathways and several mediators within those pathways to target for improving the success of CAR T-cell therapy. We've shown that CD19 CAR T-cells in advanced leukemia can expand dramatically in vivo. They could also eliminate high volume and bulky disease, and they can persist. And so we've recently published some of this, and I really have to update this slide, but with the product that we use, the CD19 CAR T-cell product, we've documented persistence in some of the earliest patients whom we've treated at Penn of over a decade, which is rather remarkable. And long-term B-cell aplasia accompanies these responses, implying that the persisting CAR T-cells really remain functional. And we also have some direct experimental evidence of that as well. And so in chronic lymphocytic leukemia, really the key, I would say, generic correlates of response are that patients who respond to the therapy, they display, again, this dramatic in vivo expansion of CAR T-cells, usually within the first 2 weeks following infusion. And then that's followed by a log normal decay of the product and the proliferation of the product with complete and often persistent eradication of the leukemia cells. And then, the infused CAR T-cells persist long-term, as I mentioned, in the responding patients. And then finally, the persistent CAR T-cells, again, are polyfunctional, which I'm not showing here, but that implies that they're actively perhaps maintaining this long-term B-cell aplasia that we're documenting. A little more on these retrospective studies. So we and others have observed that some CAR T-cells really proliferate differentially in patients, and these can form expanded populations. And so lentiviral or retroviral vectors are the typical transgene delivery methods that we heard about in the first talk, and these viruses randomly integrate into the genome to encode that CAR transgene. Insertional mutagenesis, though, sometimes alters host gene cell activity so that the locations of the integration sites, so where the CAR lands in the expanded clones, really marks genes and pathways that modulate, hopefully favorably modulate, right, CAR T cell growth and also anti-tumor potency. So I find this to be very useful for identifying modulators that can be targeted for the success of CAR T cell therapy. And I would say, you know, a prime example of this is what we published a couple of years ago. And so this was a single case study of a 78-year-old patient who relapsed through multiple conventional treatment regimens. He was infused with CD19 CAR T cells, and this patient had a delayed expansion of his CAR T cells, which was quite unusual. But after this delayed expansion, or following this delayed expansion, he experienced very robust cytokine release syndrome and tumor lysis syndrome. And essentially, by deep sequencing, we couldn't detect any of the leukemia clones. So it was a remarkable delayed response. And upon investigation, we traced this response to a single CAR T cell with a disruption in a tumor suppressor gene, a known tumor suppressor gene known as TEP2. And the cell expanded massively in vivo. I think it underwent, if I remember, something like 30 population doublings and completely wiped out his leukemia. And he's still in remission and doing remarkably well to this day. So from a single cell, and I think that really underscores the power of the CAR T cell approach if we can optimally program CAR T cells. So the big question, as I alluded to, and what, you know, we want to see as a field, I think, is a clear demonstration of efficacy of CAR T cell therapy in much more common and challenging solid tumor sort of indications. And so what factors do we really have to consider that can influence the success of CAR T cell therapy for solid tumors? Well, once CAR T cells infiltrate into the tumor bed, they're faced with major barriers that tumors often employ to blunt immune surveillance. Moreover, you know, when we infuse these therapies systemically, CAR T cell homing to the tumor sites is going to require, you know, the right cytokine chemokine gradients and the right receptor and anti-receptor interactions. And the degree of expansion of these cells is going to be greatly influenced by preconditioning of the patient before adoptive cell therapy. But as I said, even when the cells could get into the tumor, they're faced with a whole host of immunosuppressive barriers that are listed here and operative within the solid tumor microenvironment. So I had a case study here to just illustrate this, and maybe in the interest of time, I'll kind of sum this up quickly. This was a 63-year-old patient with advanced mesothelioma who progressed through multiple biologic and chemotherapeutic treatment regimens and also radiation. And he decided to enroll in one of our solid tumor CAR T cell immunotherapy trials with the product targeting mesothelioma. And so we did see a rather robust, for at least the solid tumor indication, expansion of the CAR T cells in vivo. And what was very interesting, as you could see here, there was even preferential trafficking of the CAR T cells to one of the solid tumor sites that we biopsied. But despite this, and despite a high level of CAR gene marking at the tumor site compared to the peripheral blood, still the CAR T cells were essentially shut down in that solid tumor microenvironment, and we really didn't see local proliferation or elaboration of effective anti-tumor activity. Okay, and another big problem that I wanted to mention that, you know, we have to contend with in the CAR T cell therapy field is adaptive resistance. So CAR T cell based therapeutic approaches for solid tumor indications also need to address how to increase T cell persistence and, again, anti-tumor effector function in this suppressive tumor microenvironment that actually gradually upregulates immunosuppressive barriers due to adaptive resistance following adoptive transfer of the CAR T cells. And so that's illustrated here in our trial of epidermal growth factor variant 3 directed CAR T cells in glioblastoma. And so what was really notable about this clinical study is that following infusion of those EGFRV3 directed CAR T cells, it seemed that from multiple patients and from multiple post-infusion sites, we saw upregulation of a number of immunosuppressive barriers following adoptive transfer of the CAR T cells, such as IDO, PD-L1. We also saw more regulatory T cell localization within these tumors, implying that perhaps the tumor is evolving in response to that selective pressure by the CAR T cells. So this is definitely going to be a problem. So I just want to sum up maybe some of the valuable lessons that we and others have learned from CAR T cell therapy of solid tumor indications. Despite trafficking of CAR T cells to the tumor site, some degree of initial proliferation and elicitation of effector activity, clinically meaningful responses are rarely observed in these indications. And then anti-tumor potency is frequently limited by lack of substantial expansion or survival of the CAR T cells once they hit that solid tumor microenvironment. We do, in some cases, see significant decreases in targeted antigen expression, suggesting that there is on-target, on-tumor activity, but this is often transient. And this also, I think, highlights that antigen loss and heterogeneity is a significant barrier to successful therapy. And finally, on-target, off-tumor toxicity reported in some trials, irrespective of very low antigen levels. So I think this indicates that we can get there with CAR T cells and treating some of these solid tumor indications. In the interest of time, I don't know, Miriam, if I have, I wanted to talk a little bit about universal CAR T cell products, but perhaps I can, leave that to you so that the session doesn't go over. And with that, thank you very much for taking the time to listen to my talk. And I'm certainly happy to take any questions, maybe not now, but during the panel discussion, again, in the interest of time, so that we're sticking to this tight schedule. Thank you very much for having me. And again, it was an honor to be here and share some of our work with you. Oops.
Thank you so much, Dr. Fraietta. Fascinating talk, and the solid tumor field, of course, is a totally new challenge compared to the treatment of B-cell hematologies, and I hope that the field is moving in the right direction to enable access for patients to CAR therapies in solid tumors. But looking at the hurdles, maybe more research, more evaluation is required. And I'm sure we discuss more about it in the panel discussion. Our next speaker is Dr. Khaled Al-Sayegh. He's an associate director and research scientist at the King Abdullah International Medical Center, Research Center at the National Guard Hospital. Dr. Khaled received his PhD from the Medical College of Virginia in Virginia Commonwealth University. He is specialized in human and molecular genetics, and he did postdoctoral training at the Institute of Advanced Biomedical Engineering and Sciences at the Women Medical University in Tokyo. His research interest focuses on the important aspect of bridging the gap between induced pluripotent stem cells technology and clinical applications. He works on bioengineering robust and clinically safe induced pluripotent stem cells, derived cardiac tissue, as well as on improving hematopoietic differentiation of pluripotent stem cells. Thank you so much. We are very much looking forward to learnings about the Saudi Bank of Induced Pluripotent Stem Cells.
Thank you very much, Dr. Mariam, for the nice introduction. Good evening, everyone. Thank you all for attending. Today, I'll be speaking to you about our effort at KMARC for iPS cell banking. I started with a few slides to present to you a brief background for people who are not in the field of stem cells, but what iPS cells are. Basically, induced pluripotent stem cells, or pluripotent stem cells in general, they have two defining characteristics. They have the capacity to self-renew in the lab indefinitely. You can culture them forever, basically. They will symmetrically divide. And they can also be differentiated in the lab to any cell type found in the human body. So our first encounter with pluripotent stem cells basically is at the human embryonic stem cell stage. And these cells are obtained from day 5 blastocyst stage embryo. So what you're looking at in this slide basically is an in vitro fertilization process. You see day 1, the pronuclei of the mother and the father. As the embryo starts dividing, by day 5, we reach the blastocyst stage. Basically, at this stage, the embryo is made of an outer layer called the trophoectoderm, which will basically become the placenta. And you have the inner cell mass, which is a clump of cells inside that will later solely contribute to the whole human fetus with all of its complexity, all of the systems found in that human fetus. So it's very intuitive to appreciate the potential of these cells. And indeed, in 1998, James Thomson was the first to isolate these cells, to isolate human embryonic stem cells and to culture them. And after this milestone in stem cell research, many papers have came out where they have differentiated these cells into functional cells like cardiomyocytes that are beating, or insulin-producing cells that can sense glucose levels, dopaminergic neurons, and so on. Sorry. Yeah. So, however, the stem cell culture, human embryonic stem cell research comes with its downside. Basically, these cells are problematic to be used from the ethical aspect, because these cells are, to derive these cells, we need to destruct the human embryo. And for that reason, these cells, this destruction will cause these cells to lose these embryos, and that could be problematic in some cultures and some religions. And therefore, in 2007, a major breakthrough was achieved when iPS cells were derived by overexpressing a set of genes that are known to be highly expressed in human embryonic stem cells. And then later, these cells were indistinguishable in terms of pluripotency from human embryonic stem cells, and they could be differentiated to any cell type from the human body. And the nice thing is, what's interesting about these cells is that these cells are derived from the patient, him or herself, so you don't have the chance of immune rejection. And the other thing is that these cells does not involve the destruction of human embryos, so you evade the ethical problems associated with the derivation. And lastly, these cells are as pluripotent as human embryonic stem cells, so basically they are as useful. And for that, of course, Shinya Manaka was awarded the Nobel Prize in 2012. Again, the way these cells are derived, basically we overexpress these genes in somatic cells, and this will turn them into, reprogram them back to an embryonic-like state. You get these colonies that are indistinguishable in terms of morphology to human embryonic stem cells, and they can be differentiated later to any type of cell, like beating cardiomyocyte, insulin-producing cells, and so on. And interestingly, within only 7 years of their first derivation in 2007, in 2014, the first in-human clinical trial was started in Kyoto University by Masayo Takahashi. And in this clinical trial, they used age-related macular degeneration patients that were treated by iPS-derived retinal pigment epithelial cells. You see these tiny cell sheets on the right-hand side. These were developed from or derived from iPS cells, and then were later transplanted in the back of the retina after the new vascularization was removed. And this was compared in that clinical trial with the standard anti-VEGF therapy for this type of disease. And these cell sheets have shown their superiority when compared to the standard therapy. And the other clinical trial that is ongoing now in Japan, also in Kyoto University by Jun Takahashi, is to treat Parkinson's disease patients by converting iPS cells to dopaminergic precursors, and then are intracranially injected into the patients. And also another clinical trial that is ongoing. As you can see, this is from Nature News. It's in 2018. This clinical trial is already active in Osaka University, in which they are treating heart failure patients with iPS-derived cardiomyocyte cell sheets that are transplanted on the epicardium to provide this mechanistic support, like we saw in Dr. Larson's presentation yesterday. Another clinical trial also to treat spinal cord injury with iPS cells derived neural precursors is also now ongoing. All of these clinical trials we are eagerly waiting for the first reports that will show their results. But what's interesting is that all these clinical trials that I spoke of have all transitioned to allogeneic therapy instead of autologous. And there's a good reason for that. If you saw the first clinical trial that I spoke of, the age-related macular degeneration clinical trial, the cost per patient, if you want to generate autologous cell therapy, is around $1.2 million. This is to treat a small organ like the eye. But when we go to a larger organ like the liver or the heart, the cost is going to substantially increase. And like one estimation was on liver grafts that were derived from iPS cells, mentioned that these cells could cost up to 10 million cells to be bioengineered. So the patient, because all of this will, we generate the iPS cells from the patient, and these iPS cells are gonna go through rigorous testing to ensure their safety. They are free of any oncogenic mutations. And again, we're going to differentiate the cells to make sure that we can generate functional tissue from them. So all of this, the patient sometimes does not have the luxury to wait for such a long time, and the cost could be, again, prohibitively expensive. So the alternative and why the reason why these clinical trials are moving towards allogeneic is basically to reduce the cost and at the same time reduce the time and wait by having off-the-shelf cell therapy that is optimized for the population. And the way we do that is by HLA hemoleukocyte antigen matching. So the proposed mechanism for that is basically we have what is called a universal donor. Is we go through the population and we identify the HLA distribution in the population. And then if we can find homozygous donors who are homozygous for the most common HLA haplotype, these donors can match the large population and large percentage of the patients that are in need. So this prompted us to investigate the feasibility of HLA-based banking in Saudi Arabia. So the first step was to survey the population in Saudi Arabia and see the HLA frequency and distribution in that population. When I first joined KMARC in 2015, KMARC was just beginning a great initiative called the Stem Cell Donor Registry. It's a registry that is targeted towards providing donors for cancer patients and patients with hematological diseases. What they did is that they collected a big number of that registry and sequenced the HLA in high resolution, as you can see on the right side table. And these are the frequencies shown here. So we accumulated these haplotypes, and we found that if we reprogram 7 of these donors, we can cover more than 10.3% of the Saudi population. So this is the paradigm of the proposed treatment, is that we can find this universal donor, and then we're going to focus on hemoleukocyte antigen locus A, B, and DRB1. And as you can see, the top 2 patients, they are compatible, but at the DRB1 locus, the last one is not matching. So at KMARC, we generated our first IPS line, and we were interested in making the cell line as clinically relevant as possible, because KMARC is taking the direction of establishing its GMP facilities and cell processing centers. And we want to work in parallel until we develop these therapies and are ready for application later on. So we used simply, instead of skin biopsy, something as invasive as that, we used a 10 ml peripheral blood sample. And from that, we isolated erythroid progenitors. And these cells are free of any genomic rearrangement usually, and they are more abundant in blood than CD34 positive cells. And these cells are pluripotent. As you can see, they have the pluripotency markers. And at the same time, they are karyotypically normal. So at KMARC, we have 2 projects basically that we want to, that's about to start soon, is the generation of iPS-derived biological makers or cynoatrial node cells from these iPS cells. On the other hand, we have another project that aims to generate natural killer cells or CAR NK cells to target B-cell lymphomas. So in this project, in collaboration with KAUST and King Faisal Hospital, we're going to use single-cell RNA sequencing we have these beating cardiomyocytes here. I don't know if the video can be turned on in this slide by the organizers. And also, we want to do single-cell RNA sequencing from the primary heart tissue obtained from explanted heart of patients undergoing heart transplantation. The single-cell RNA sequencing is going to allow us to see how these generated iPS products can compare to the real thing. And that will, the end goal of this is to develop biological pacemakers that have pacemaking ability. All right. The other project by Dr. Abir Sufyani is to generate these natural killer cells. So basically, we're going to reprogram the patient or the donor cells, and then we're going to engineer to have the CAR CD19 antigen, and then differentiate it to natural killer cells. And we're going to also employ here single-cell RNA sequencing to assess the comparison between these cells and natural killer cells obtained from the donor. And the ultimate goal of this, again, is to have off-the-shelf therapy to one day be used on Saudi patients. And again, we want to contribute to and collaborate with other institutes in the kingdom who are interested in developing cell therapies derived from these iPS cells. So we're going to distribute this universal cell lines to other collaborators who are interested in developing iPS-based cell therapy. So some collaborators might be interested in developing insulin-producing cells from these. So this collaboration is going to push forward this iPS-based therapy. The other arm of that project is to bank patient-derived iPS cells. So as you know, iPS cells can also be used for drug discovery. If we have a patient or a disease in which the tissue that is affected is inaccessible while the patient is still alive, what we can do is we can generate iPS cells from these patients and differentiate it to the lineage of interest and examine that tissue for difference and do drug screening. So this is basically the plan is to, because King Abdulaziz Medical City has around 2,500 beds capacity. So we generate iPS cells from these patients, and then these lines could be sold and distributed for pharmaceutical companies and academic universities to do drug screening and understand disease mechanisms. And this can also, the monetization of this could be really helpful. One project led by Dr. Dua at KMARC is to study a disease that is caused by SLC mutation. We're going to generate iPS cells from the patient and from the normal sibling, and then differentiate these to generate midbrain organoids that can later be used for drug screening and understanding disease mechanism. So I'd like to thank all the collaborators and KMARC, Dr. Askar, Dr. Majd Al-Fadl, Dr. Dunya Jodot. Thank you all very much.
Thank you so much, Dr. Khaled, for that really fascinating talk and introducing us to the opportunities iPS cells may bring in the future. Other questions for Dr. Khaled? If not, I would like to introduce our next speaker, Dr. Amin Ayitu. He is a professor and chair for targeted therapeutics at the Imperial College in London. He has completed his PhD at the University of Liège in Belgium, and during his PhD work, he acquired extensive experience in gene delivery technologies using eukaryotic viral vectors. He completed his postdoctoral training at the MD Anderson Cancer Center in Texas, where he gained experience in bacteriophage, guided gene delivery, and phage display technologies in vitro and in vivo. Importantly, he designed a novel hybrid phage vector for targeted gene therapy transfer. In 2008, Dr. Ayitu established his research team at the Imperial College in London, where he became a senior lecturer in 2013 and a professor and chair of targeted therapeutics in 2019. His research team has become a leading authority in phage-guided gene delivery technologies, focusing on optimizing and developing superior phage-derived vectors for nucleic acid delivery to human diseases, including DNA vaccines. His leadership in the field has resulted in various awards for his research, numerous high-profile publications, and more than 6 patent applications, which also have contributed to the creation of 2 startup companies in the US. He is also a scientific co-founder of a spin-off company at the MIT in the US. He has received a royal decoration in 2015 by His Majesty, the King of Morocco. I welcome an expert in the field of development of bacteria viruses, bacteriophages, into a delivery technology for therapeutic genes and DNA vaccines against cancer and human diseases. The floor is yours.
Thank you very much for this nice introduction. And I would like to thank the organizers for inviting me to this wonderful conference. Can I have my first slide, please? Can I have my first slide, please? Yeah. I don't see it. Could you show the slide, please? I don't see it on the whole screen. Yeah, I think there's a problem with slide. I don't see the slide on the screen. Can you show it on the screen, please? Okay, yeah. So let me show. I don't see my slides on the screen. Yeah, we can get started. So today I will present what we have done over the last few years to develop these bacteria viruses as vectors for targeted gene therapy of cancer. Can I have the next slide? Yeah, so as you can see, gene therapy has been around for a long time. It's actually the delivery of therapeutic genes. Gene therapy was initially conceived to treat inherited diseases, but today more than 67% of gene therapy clinical trials are designed to treat cancer. Because of course, cancer is a major cause of mortality in the world. And glioma, which are deadly brain tumors, were actually the first disease to be treated in the clinic by gene therapy, but clinical trials failed because of the vectors used at the time. Actually, to deliver therapeutic genes, you need vectors. Can I have the next slide? And human viruses have been mostly used because they can enter human cells as part of their infection process. So lentiviral vectors, retroviruses have been used, adenoviruses have been used, the herpes simplex viruses have been used, and adeno-associated viruses have been used to derive vectors. And as you know, the AAV, adeno-associated virus, has become the most popular virus to derive vectors for clinical trials. And the principle of making vectors is the same. What you need to do, you need to make these viruses replication deficient. It means you need to remove the native genes, like the case here of AAV, adeno-associated virus. You need to take out the rep, which is the gene for replication, and the cap, which is the gene for the capsid, and replace them with the mammalian transgene cassette, which is the CMV promoter, commonly used promoter to express the therapeutic gene on poly-A sequence. That's the principle of making vectors from viruses. Can I have the next slide, please? So As you know, gene therapy has been around for a long time. But for many decades, the big question why gene therapy hasn't delivered the expected outcomes in the clinic, because gene therapy has faced many problems, but mainly systemic delivery. That's why the majority of clinical trials of gene therapy, they are injected actually locally, because there is a problem with systemic administration. Local administration can show proof of principle, but real clinical benefit can only happen with systemic administration. For instance, if 90% of cancer patients, they die because of metastasis, and metastatic cancer requires systemic administrations. You can't treat all these metastases with local injections. I'm showing here the example of this mouse that I treated a long time ago. This is systemic administration. It's an intravenous administration of an adenoviral vector carrying the luciferase gene. This is an imaging, so I injected the virus into the tail vein. This mouse had a tumor, Kaposi sarcoma. The virus could deliver the gene everywhere, but not in the tumors. You can see they get trapped by the liver, by the kidneys. These viruses have broad tropism. That's why there is a problem. I mean, that's why systemic administration, systemic gene therapy has had limited success in the clinic. And one solution is to target them, to target these viruses. But what you need to do first is to ablate the native tropism, which actually which has been a problem. It was not a success. That's one thing. And another problem is the repeated administration, because to achieve a therapeutic response, you need to repeat the administrations. But these viruses are immunogenic, so they block the repeated administrations, which actually hinders the successful complete response in individuals. Of course, gene therapy has faced other problems like the packaging capacity, the cost, production to GMP standard, scaling up the production. Can I have the next slide, please? So now I'm going to try to show you what we have attempted to do to solve at least some of these problems. So what we did, instead of using human viruses, we turned to other viruses, bacteria viruses, named bacteriophages or phages. If I ask you the question, before the discovery of antibiotics, how bacterial infections in humans were treated? By these viruses, by bacteriophages. They do not infect human cells, they do not infect human tissues, so we can deliver them systemically, intravenously, without a problem, because they wouldn't get trapped in the liver and other tissues. They were discovered in 1895, and then later they found that these bacteriophage kill bacteria. That's why they used them before the discovery of antibiotics. They used them to treat bacterial infections, but then after the discovery of penicillin, they were discarded in the West, but some countries in East Europe continued to use them. But now in the West, their interest is growing back because of the antibiotic resistance. Can I have the next slide? For instance, in this paper in Nature Medicine in 2019, this was a clinical trial to save this 15-year-old cystic fibrosis patient. She had an infection with Mycobacterium, which is resistant to antibiotics, but then they treated her with a cocktail of bacteriophage, which actually saved her life and cleared the bacterial infection. And in 2006, the US Food and Drug Administration approved the use of bacteriophage as antibacterial food additives to prevent the growth of Listeria, Salmonella, and E. coli. The point I'm trying, next slide, the point I'm trying to make here is that these viruses are safe. If you want to come up with a new vector, new treatment, new gene therapy approach, you need to take into account two crucial considerations. First, safety. They are safe. They have been given to human. Second, efficacy. That's why we decided to use them. We can deliver them systemically. But the big question, how can you make a bacteria virus enter into a human cells? What we did, next slide, we used the phage display technology, which won the Nobel Prize in chemistry in 2018. The phage display technology consists on the display of ligands. And you modify the capsid of phage. You just express ligand to make these phages bind to specific receptor into cancer, for instance. Then they become targeted. Next slide. So we used the filamentous bacteriophage here. It's just a virus with coat proteins covering the genome. There is the gray protein, the major coat protein, and you have two minor coat proteins on each side. Next. So you use the phage as first what we did, we used the phage display technology to display this ligand, RGD4C, because it binds to this receptor alpha-5 beta-3 integrin, which is expressed in cancer, but minimally present in the healthy tissues because we want to target cancer. That's for the entry. So the whole, as soon as the virus binds, it gets internalized, and then the genome of bacteriophage gets released, but it doesn't work in human cells. It only works in bacteria. So to express the therapeutic gene, what we did, we inserted within the genome of phage the genome of the human virus, AAV, because both are single-stranded genomes, so they are compatible to insert into each other. But the big challenge was whether we could produce this in bacteria. So we take the hybrid genome, we transform the bacteria, then we were able to package the hybrid genome of phage and AAV by the capsid of bacteriophage in bacteria. Next. Then if you take this, we name it AAV phage, if you add it to a panel of tumor cells, if you don't have the ligand RGD that allows entry, there is no GFP delivery. But if you display the RGD on the capsid, it gets inside the cells, it delivers AAV, expresses GFP. Next. The same thing in animals. If you take this phage, you inject intravenously into mice with tumors, what happens? It gets cleared. If you use a ligand that doesn't target cancer, it gets cleared. But if you use the RGD on the capsid, the RGD will guide the phage to these receptors integrins into the tumors, enter into cancer cells, deliver the AAV to express the luciferase gene. That's what we want to achieve with target. You see, there was no delivery to the healthy tissues. And another group used this technology to deliver the tumor necrosis factor alpha, TNF alpha for immunotherapy, cytokine gene therapy, because TNF alpha has strong anti-tumor activity, but it's toxic. It has to be targeted. So when we cloned into our phage, we delivered IV into mice, intravenously into mice with tumors, they were able to recover TNF-alpha expression into the tumors, but no TNF-alpha was detected in the healthy tissues, the brain, muscle, lung, kidney, and heart, and other normal tissues. And this is what you want to achieve with targeted. In chemotherapy, is the other way. You get 5% in the tumors, the rest is spread all over the healthy tissues. Next. So if you make sections of these tumors, what you see that actually you have delivery. This is GFP delivery to the blood vessels in red and the tumor cells because they express the receptor. If you replace GFP with TNF-alpha, then you blew up the blood vessels on the tumor cells. So if you remove the RGD, nothing happens. So you really need target. You need that ligand to guide the phage to these tumors. Next. What I would like to show you here is suicide gene therapy because it was the first one to be tested actually in the clinic, in clinical gene therapy against glioma. What you do, you deliver the thymidine kinase of the herpes virus, you give ganciclovir, it gets phosphorylated by TK, it destroys tumor cells. So that's what we did. So targeted suicide gene therapy with our virus completely stopped the growth of these tumors in mice as well as in rats. Next. So then we did, in collaboration with a group from the National Cancer Institute in the US, we did a safety study in larger animals. These are pet dogs with huge tumors. These are dying dogs, and the owners, they bring them to the NCI. So the vets, they did like phase 1, dose escalation, but we never reached an MTD, maximally tolerated dose, because phage is safe. We know phage is safe. And then the vets said, because these tumors are huge, we have to repeat the administrations. But are they safe? Then we repeated once a week administrations for months. They were safe. They were not toxic. And then the big question is that because phage is immunogenic, there are IgGs against phage capsid after 2 weeks. But do they block delivery? Next. Then we did a biodistribution because after 8 weeks, 8 injections, we could still see the phage in red in the tumors, but no phage was detected in the healthy tissues. It means these antibodies against the phage, they don't stop the phage. Unlike the human viruses, the first injections will hinder the efficacy of the successive injections. And this selective accumulation of the phage in the tumors of the dogs resulted in selective expression of TNF-alpha into the tumors, not in the healthy tissues. So this technology is bringing TNF-alpha back to the clinic. Next. Then we saved dogs. I'm showing the example of these dogs with huge tumor. It was a fibrosarcoma. So we treated with, you have to repeat, we repeated for 2 months, once a week. So from after 4 weeks, from 15 centimeter, the tumor shrinked to 8 centimeter. And after a total of 8 weeks, IV injections, the tumor was completely eradicated. Next. So in the next part of this talk, I will show you what we have done for vector development. That's the challenge, the problems that gene therapy has faced, because people rush to the clinic. We didn't want to rush to the clinic. We wanted to improve that, because phage has evolved to infect bacteria with no optimized strategies to deliver genes to human cells. For instance, in this publication, if you look at human viruses, they get internalized into the endosomes, but they escape from the endosomes to avoid getting into the lysosomes, to avoid degradation by the lysosomes. But bacteriophage doesn't have tools to do that. This EM shows that the bacteriophage is in the early endosomes, in the multivesicular bodies, but the majority of phage ends up in the lysosomes because it cannot escape. That's a major problem. We had to solve that. If you give chloroquine, which disrupts the endosomes, then the phage can escape and increase gene delivery. Next. So in this publication, we used direct devolution. Actually, what we did was how we can transfer the strategies developed by human viruses to the phage capsid to make them escape from the endosomes. So if you look at this phage on the P3 minor code protein, the RGD, we displayed the RGD. But then what we did, we wanted to display peptides from human viruses, and that's why we inserted a recombinant major code protein on which we could display endosomal escape peptide, which allowed the phage to escape from the endosomes, as you can see, and increase gene delivery in vitro as well as in vivo. Next. So here I will show you what we have done to treat glioblastoma, because these are deadly brain tumors, the first to be treated in the clinic. The majority of patients, they die within, 99% of patients, they die within 3 years. So we wanted to treat glioblastoma because phage can cross the blood-brain barrier. It's a major challenge in treating brain tumors and brain disorders. Next. What we did here, we instead of using CMV promoter, we used the promoter that transcriptional targeted that is active in glioblastoma. It's actually associated with glioblastoma resistant to chemotherapy. So if you treat the cells with the phage, with this promoter, you give them low dose temozolomide, then you activate the therapeutic gene from this GRP78 promoter. Next. So in this publication, that one viral in the public, actually, we take human tumors from patients, we insert them with phage packages, both the genome of phage and the AAV. What we did was how to generate phage that can package AAV only. So the way to do that in bacteria, so we take the AAV, we're talking here about instead of packaging AAV genome by the AAV capsid, is to use the phage capsid. We have to do this in bacteria. So you take the phage, the AAV genome, you transform the bacteria, but you need to make it replicate. Then we inserted an origin of replication of phage, then the AAV starts to replicate in bacteria. And then we infect with the helper phage to provide the capsid. Then we manage to produce small particles with the phage capsid and AAV genome only. But the advantage is that we obtained a dramatic increase of gene delivery. Next. And you see, these are small particles, actually. And then we use them, we carried out a comprehensive analysis of gene delivery to compare to the large particles. It's interesting when you reduce the size, when you treat like primary tumors in vitro from patients, from glioblastoma patients, the large particles have difficult to deliver, while the small particles could deliver to all these tumor cells and stem cells and tumor stem cell sphere in vitro. Next. Just to show you an application for CRISPR, because we reduced the size of these particles, we increased the packaging capacity, then we could fit in CRISPR actually, and to deliver Cas9 expression to the human lung carcinoma, up to 70% of the cells were positive for Cas9. And then we could knock down the expression of the mutant P53. This mutation is present in 50% of lung cancer. Next. Just I will finish now with immunotherapy. Just we used it for immunotherapy. Just to give you an example of interleukin 15, it's a cytokine. It's in the clinic. They give this protein to the patients. There is a response, but there are problems of short half-life and renal clearance. So we decided to do this as gene therapy. So we inject mice with tumors. These are IV injections with increasing doses. You can see that all the doses generated expression of interleukin-15 in the tumors, but not in the healthy tissues, because we overdosed the mice. We didn't have any safety issues as confirmed by the LDH, which remained its cytotoxicity biomarker, which remained intact in the serum. And this immunotherapy can be a powerful tool, actually. You can see that the tumor regressed as early as day 5 after treatment with a targeted vector compared to the control. Because we can repeat the administrations, we could then, after a few weeks, we could completely eradicate the tumors, 50% the animals. We saved 50% of the animals that were still alive even 1 year later. Do I still have a couple of minutes? I can finish with the vaccine, just a couple of slides. Can I have next slide, please? Just to finish that, with this slide, that bacteriophage has been used as a vaccine delivery system because of the phage display technology. You can display antigens on the surface because the phage gets uptaken by the antigen-presenting cells, and because the phage can interact with MHC1, MHC2. And in this publication, we showed that the phage can cross the gastrointestinal tract. So phage can be given as oral vaccine. That's why we used it in cancer vaccine. Next, cancer vaccine consists of vaccine that can induce an immune response against the tumor-associated antigens or tumor-specific antigens, but the tumor-associated antigens, they are also expressed in other tissues, and the tumor-specific antigens, they get down-regulated by the tumors. That's why we used a novel approach, next, in which we decided to deliver foreign antigens to the tumors that could be viral. The idea is just to inject the animals or patients with any vaccine. It could be COVID vaccine, flu vaccine, malaria vaccine, as long as this vaccine can induce an immune response against the tumor. So then if you take these animals, you establish tumors, they are vaccinated against, for instance, malaria. What you do, you take the phage, you inject IV to deliver the antigen into the tumors, then you induce an immune response against the tumor. If you take the splenocytes from the vaccinated animals, you can see the if you mix them with tumor cells, they don't kill them. But if you mix them with tumor cells and you give the phage with the antigen to tumor cells, you destroy them. Same thing happened in animals. The tumors, they grow in vaccinated animals. But if you give the phage to deliver the antigen to the tumors, then you induce an immune response against the tumors. Next. So conclusion, just since the dog study in 2009, we uncovered limitations of these particles. We designed strategy to improve them. Then we are ready for clinical trials now. We believe we have the vector to enter clinical trials. Next. If you look at other strategies used, the advantage of our strategies is that it's cost effective, it's targetable, we can repeat the administrations. We don't need cold chain transport. The phages are stable at room temperatures. So our phages can actually solve various problems that gene therapy has faced for many decades. Next. So as you mentioned earlier, so this has generated a couple of companies in 2013, the first generation of phage, which is going to clinical trials, and in 2021, another company with a group from the MIT. Next. And I would like to thank my group and all the students who have worked hard and the funders in order to advance this technology toward clinical trials and commercialization. Thank you very much.
Thank you so much for that truly fascinating talk and presenting us insights into something which really could be a game changer in the treatment of patients. In the interest of time, I suggest that I would start with my presentation. So my name is Miriam Fuchs. I'm a global therapeutic area lead at Novartis in regulatory affairs, cell and gene therapies. I'm a PhD by training. I worked on signaling at the Max Planck Institute in Munich, and I did a postdoctoral fellowship at the Dana-Farber Cancer Institute in Boston, again on signaling in cancer biology. And I joined the biotech and pharmaceutical industry approximately 20 years ago. And I spent the last years in a really outstanding team developing tisagenlecleucel for the benefit of patients. Let me just see how I work the slides. This is my disclaimer. Yeah, so our journey for tisagenlecleucel, also known as CTL019 or Tisacel, started in August 2012 when Novartis entered into a licensing and development agreement with the University of Pennsylvania to develop CAR-T therapies. At this time, the first patients were successfully treated with CTL019, including Emily Whitehead, who had been the first pediatric patients ever treated with a CAR-T cell therapy. Shortly after initiation, the collaboration with the University of Pennsylvania, Novartis purchased a cell manufacturing facility in New Jersey to enable manufacturing for global registration studies. During the course of the tisagenlecleucel development, we used the available tools for an early and close interaction with the regulators, like breakthrough designation with the US FDA and prime designation in Europe with the EMA. And these designations are unique opportunities for an increased dialogue with health authorities and are really helpful in defining the development and registration strategy for innovative products like cell and gene therapies. And we were the first company to run truly global pivotal studies with CAR-T therapies. In August 2017, Tisacel received approval from the FDA for an indication to treat pediatric and young adult patients with relapsed refractory B-cell ALL. And it was the first CAR therapy ever receiving regulatory approval by a health authority. In 2018, we were able to expand the use of Tisacel to include adult patients with relapsed refractory diffuse large B-cell lymphoma after 2 or more lines of therapy as a second indication. But equally important, we were able to gain regulatory approvals for both indications in Europe and many other regions, including Saudi Arabia, to enable patients globally to access CAR-T therapies. We recently announced that the ELARA study, which is investigating Tisacel in third and later line relapsed refractory follicular lymphoma patients, met its primary endpoint, and I will share some data on that indication. For me, the promise of CAR-T cell therapies is symbolized by Emily Whitehead, who had celebrated in May this year 9 years of cancer-free remission. I will skip this slide because we had a very eloquent introduction to the mechanism of action of CAR-T therapies. So the logistics of Tisacel is complex. We heard about it already, given the shipment of cryopreserved leukapheresis from the apheresis center to the manufacturing site, which often is outside of the country where the patient is treated, and then the delivery of the cryopreserved final product back to the clinical site. And of course, all steps are requiring a close control of chain of identity, which is absolutely crucial for an autologous product. The manufacturing of Tisacel itself is sophisticated, including transduction with a replication-deficient lentiviral vector to stably introduce the transgene encoding for the CAR. The manufacture itself lasts around 9 to 10 days, followed by a final product quality release testing, leading to an overall time for manufacture and release of around 22 days. At the clinical site, the patient is receiving lymphodepleting chemotherapy to enable expansion of the CAR-T therapy before administration of Tisacel. And CAR-T therapies are indeed living drugs, and they are supposed to be a one-time treatment. Sorry, I'm moving the slide. So, however, not only the manufacturing and the logistics around autologous CAR-T therapies are very different from what we face with small molecules and biologic. The interactions at the treatment center itself are equally complex, and a close collaboration between multiple stakeholders at the clinical centers, as visualized in this slide, is instrumental for the successful management of CAR T therapies. We were very dedicated to bring Tisacel to patients around the world, and we have now established a global reach with a presence in many regions, as visualized on this map, with access to Tisacel in 330 centers around the globe, and an intention to continue to expand the global availability. A global reach also requires a large manufacturing footprint to provide reliable capacity for manufacture of CAR T therapies for clinical trials as well as for commercial setting, and to enable a geographic reach also from a logistics perspective to facilitate the logistics. We have established 7 facilities around continents to manufacture CAR T therapies, either Novartis owned or working with contract manufacturing organizations. The clinical development of tisagenlecleucel was linked to important learnings on our side, and I would like to share some of the unique features related to the development of CAR T therapies, which to some extent can be extrapolated to advanced therapies development in general. From a clinical trial perspective, the impact of manufacturing an autologous product individually for every patient on the study conduct itself cannot be underestimated, including, for example, the timing between patient enrollment and the infusion of the therapy. Given the often progressive nature of the disease investigated, patients may need to be stabilized with bridging therapy while the product is manufactured. The execution of global multicenter studies requires dedicated onboarding and training of the clinical sites for the management of the CAR T therapies, but also for the local logistics. The logistics for a global study, especially if you have only one central manufacturing facility, need to be established, as well as a close control of chain of identity. And often limited manufacturing capacity is faced in early phases of the program, and that may pose a challenge, as well as adjustments to the manufacturing process, which may very well happen in the early phases of development. Also, development programs may be condensed, especially in later lines of indications when no effective standard of care is available. And many of these advanced therapies are actually developed for rare indications. Consequently, there's a unique role of the natural history data of a disease to contextualize the pivotal study results. And indisputably, academia plays an important role in driving the innovation in cell and gene therapy space. Many early cell therapy clinical trials are initiated by academic centers across the world, as these centers are actively engaged in investigating novel approaches and technologies. From a CMC perspective, however, it is critical to consider that these therapies may be requiring iterative development and optimizing for GMP production at commercial scale, especially when initially developed at these academic centers. In respect to autologous products, we have to consider variable patient material as a starting material. We may also face lot-to-lot variability of human and animal-derived materials used in manufacture. There is potentially also limited product knowledge in the early phase of clinical development, and all these elements may pose challenges in the development of a robust commercial process. We need to understand the product critical quality attributes, the critical performance parameters. We need to establish clinically relevant product specifications, develop robust analytical tools for release. All these are critical aspects in developing a suitable commercial manufacturing process and control strategy that ensures a consistent product quality. Also, establishing reliable comparability methodologies is a critical prerequisite in the context of process improvements, which may be introduced during a clinical study, as well as to expand the manufacturing process to new facilities. The initial development of tisagenlecleucel focused on indications in pediatric and young adult patients with relapsed refractory ALL and adult patients with relapsed refractory diffuse large B-cell, both indications with high unmet medical need. And at the time of the study initiation, there were no effective treatment alternatives available. So our pivotal program was based on single-arm studies. And the pivotal data allowed to conclude a positive benefit risk profile on tisa-cel in the approved indications. However, we know the experience in real-world clinical practice is very relevant, and for CAR T therapies, this experience is documented in registries for CAR T therapies, like, for example, the registry from the Center of International Blood and Marrow Transplant Research, CIBMTR in the US, or the EBMT in Europe. And these organizations entertain registries which were originally developed to follow hematopoietic stem cell transplant patients, and they have been expanded to also follow CAR T patients after infusion. Importantly, as presented in this table, the experience in real-world clinical practice with tisa-cel is consistent with the pivotal trial experience in respect to efficacy, as shown by the overall response rates and the survival data. And the data also show that the safety of tisagenlecleucel is generally well manageable by physicians in the commercial setting. I will take a moment to speak about our experience with tisa-cel in adult patients with relapsed refractory follicular lymphoma, which presents the second most common form of non-Hodgkin's lymphoma after diffuse large B-cell and is the most common form of indolent non-Hodgkin's lymphoma. And follicular lymphoma is characterized by relapsing remitting phases and is considered incurable in advanced stages of the disease. Our pivotal single-arm study called ELARA investigated tisa-cel in patients who were refractory to 2 or more lines of systemic therapy, including a CD20 antibody and an alkylating agent. We have seen in a prior presentation the study schema for the early clinical studies, and it's similar for the follicular lymphoma study. It's a single-arm study with phases of screening, leukapheresis, cryopreservation. Patient is enrolled, potentially receives bridging chemotherapy while tisa-cel is manufactured. Prior to infusion, there is a lymphodepleting stage. Tisagenlecleucel is infused, and there are regular assessments to monitor response and safety. The primary study endpoint was met at the pre-planned interim analysis with a high complete response rate of 66% and an overall response rate of 86%, shown on the left table. And of note, the durability of responses presented in the right graph indicates a clinical benefit for many of the treated patients. And based on available data, we propose to conclude that the benefit risk profile for tisa-cel is favorable for the treatment of patients with relapsed refractory follicular lymphoma after 2 or more lines of therapy. And we will seek regulatory approval for these indications to enable patient access. Innovation does, of course, not stop with the first cell and gene therapies receiving regulatory approval. The field is now facing rapid cycles of technical and scientific innovation to provide patients with novel treatment options and to achieve better outcomes for more patients in more indications. For the CAR T specifically, the field is innovating to achieve higher response rates, overcome the mechanism of resistance, and improve the persistence of responses, as well as to expand to new indications, including solid tumors, and we have heard about it in the prior talks today. Loss of CD19 antigen, for example, is well documented in pediatric ALL as a cause of relapse. We spoke about it. Other mechanisms of resistance are also present, such as CAR T-cell exhaustion, lack of persistence of CAR T-cells, or resistance of cancer cells to killing by CAR T-cells, even when the CAR T recognizes the antigen on the surface of the cancer cell. And on the left little figure, we are presenting some of the potential mechanisms to tackle the hurdles which have been described in the earlier presentations. Targeting to antigens, for example, by applying a dual CAR T is an approach that may help to address the mechanism of resistance related to loss of an antigen. However, this would not address the mechanism of resistance due to T-cell exhaustion, loss of CAR T-cells due to limited persistence, etc. Combination strategies with small molecules or antibodies might help to overcome some of the tumor intrinsic mechanisms, and that's of course a field of intense research. Importantly, avenues to improve T-cell fitness may also improve the clinical outcomes. And to this extent, Novartis is developing a novel CAR T manufacturing platform that will serve as the foundation for the next generation CAR T therapies in our pipeline. And this novel manufacturing process results in a product in which T cells retain a less differentiated, more stem cell-like phenotype, which is thought to be an important T cell characteristic closely tied to the therapeutic potential of CAR T cells. And thus, it is expected to lead to deep and durable responses and improved long-term outcomes. We can expect that some of the improvements in the CAR T field we are seeing now may be incremental in respect to clinical benefit. However, of course, every improvement is meaningful for patients. And one of the challenges ahead of us is to define how we can best develop these innovations to assure timely patient access to these improved therapies. Going forward, a close collaboration between developers of advanced therapies, regulators, and HTA bodies, the payers, early in the development process will be instrumental in order to gain regulatory approvals and enable timely access for patients to innovation. So early engagement is critical to align on key aspects of the development program, like trial design, endpoints, patient population, but also to recognize long-term uncertainties and address them in the development program and post-authorization. Specifically, the use of real-world data, real-world evidence to contextualize pivotal data pre and post-approval will be key in these discussions. And especially in the cell and gene therapy field, there is a unique opportunity, but also a need for the use of real-world data to contextualize the clinical trial data. I would like to conclude with a deep appreciation of where the field is now, but also to emphasize the importance of a close collaboration between all stakeholders, academia, biotech, pharma, physicians, regulators, payers, as well as patients, to ensure that the pioneering work in the field and the rapid innovation cycle we are currently seeing can reach and benefit patients in a timely manner. Thank you for your attention. Are there any questions? Otherwise, I would suggest that we move to the panel discussion. Okay, so thank you for the very, very interesting presentations we heard in that session. I really enjoyed it. I would want to pose a few questions, and I would like to start with the first one. We have seen that the recent development of CAR T-cell therapy has revolutionized the field of cellular therapies. How do you see the accessibility for CAR T therapies changing in the future? What is the economic impact of these CAR T-cell therapies and the expected impact over the next few years? And I would like to pose that question to Dr. Bader.
I'm a clinician by nature, and I'm not an economist. CAR T cell has really revolutionized the way we treated patient and introduced a new idea in not doing an innovative way, but also an effective way of treating those patients. I mean, we are seeing more durable responses, and that can lead eventually to cure for some of those patients that used to be an incurable patient. In terms of economical impact, I mean, the cost of the medication of all this treatment considerably high when you compare it with other type of therapy. For example, and however, I mean, it is working. It's giving good result, but I'm not sure about the how this treatment will be readily available for patient, and how patient will have an appropriate access for this kind of therapy. In terms of economic impact, it has been estimated in US, for example, in 2026, the CAR T cell therapy market will reach up to $1.7 billion for CAR T cell that treated hematological malignancy. That's a good share of market. However, the expenditure is going to be a burden in the buyer. And for example, recently published data from Europe, for example, showing that in European countries, the expenditure will be increasing over the next few years, up to 2029, showing that cumulative impact on economic impact on those countries. For example, it has been estimated that countries such as Netherlands will expend around 1.4 billion euro, up to 1.6 something euro for a country such as Germany. That's only a direct price for the CAR T cell product itself, including hematological malignancy, and that's including myeloma products as well. So that's a huge expenditure, and of course that will be equal to a huge economic impact.
Okay, thank you so much for that assessment. My next question is directed to Dr. Fraietta. CAR T-cell therapies are now one of the most promising and sought after cancer treatments. How do you see this area evolve over the coming 5-10 years? Off the shelf, allogeneic CARs, CAR NKs?
Yeah, that's a great question. So accessibility is key, I think, in the autologous space, even though we've demonstrated that we can scale up and scale out, right, and make commercial products. Too many patients, as we've heard today, are still unable to receive their autologous products because of, manufacturing failures, or the fact that, and again, I'm not a clinician, I'm a biologist, but this is what I've seen, or they're not stable enough, right, to receive their CAR T cells. So I think allogeneic products are really the future, because we can increase patient access and have, hopefully on-demand, products for on-demand treatment. And so I think, developments in the field over the next coming years in the areas of safety, reliability and efficacy of these products, and it'll start as it is now, right, in the heme malignancy space, are really going to determine how disruptive I think CAR T cells overall can be in the broader battle against cancer. So I think in the liquid tumor indications, and we're seeing this now, we're making great progress, but in those indications, I think you need product persistence, and we haven't figured out how to do that yet or achieve that yet with allogeneic products. But I think we're getting there, and I'm a believer, and I'm very hopeful.
Thank you. Do you think that CAR NK cells, allogeneic CAR NK cells, would have a better chance than allogeneic CAR T cells?
Oh, that's a good one. That's a good one. I mean, I'm a T cell guy, and Okay, so if we turn to solid tumors, perhaps, right? But solid tumors, if you just look at endogenous solid tumors, they're not really infiltrated by NK cells. So I think we're going to have to do a little more engineering to figure out how to get them where they need to be. And they're also susceptible to immunosuppression within the tumor microenvironment. You know, you can manufacture them, but I think just like what you're doing at Novartis, we need manufacturing improvements as well for these cells. So for instance, I know if you like soak NK cells and IL-15, which I think everybody does to keep them surviving and to, you know, manufacture them, they tend to be exhausted once if you could get them to a solid tumor site. So I think there's going to be a lot of crosstalk with what we're doing in T cells. But yes, obviously, that could solve potentially, you know, a lot of issues with patient access. I think it's going to come down to, do we have to keep re-administering these? How often are we going to have issues with adaptive resistance, like we're seeing with T cells across several indications, across several centers, if we have to hit hard and heavy and keep re-administering them. So, but they're a very attractive modality for sure.
Okay, thank you for that assessment. Stem cell therapy has raised a lot of hopes, yet clinical successes in this field are still limited. We have seen some interesting data today. Dr. Khaled, what are the challenges faced in the development of the stem cell therapies? Do you think they could ever become routine clinical practice?
I mean, that's the hope, right? So the major challenges are basically to have full grasp on cell fate and be able to differentiate these iPS cells or human embryonic stem cells to where we exactly want them to be. We want to generate functional cells from these cells that resemble exactly the primary cells that are found in the adult human body. So basically is efficient and highly accurate differentiation of these cells. This is one. The second thing is scalability. I mean, with the usage of bioreactors, this have been improved. And then the other thing is the whether it's autologous or allogeneic, that's another issue. There are some countries where the genetic pool is very heterogeneous, and having these stem cell banks might not be the best option for them. But for places like South Korea or Japan, and also as I presented today in Saudi Arabia, it seems like a good direction to take. So we are now assessing the feasibility of this. And these are the major problems. Problems other than that is basically the teratoma formation was at once a big concern, but as we can see now that there are so many ways that have been developed to get rid of any contaminating pluripotent stem cells after we differentiate these cells to the tissue of interest. So that is no longer that much of a concern like it was before with all these developed techniques. So basically the cost and the full grasp and control of the cell fate of these cells is really key.
Okay, thank you so much. Similar to the stem cell therapy, gene therapy as well has still limited success cases. Dr. Aishitu, what are the common scientific and technical challenges the field is facing from your perspective?
Thank you for this very important question. Well, I can say that gene therapy is still suffering the mistakes and problems of the 90s. At that time, gene therapy caused death of patients. The best example is that of the child, J.C. Gilsinger, because at the time, scientists and clinicians rushed to the clinic to inject these vectors into patients without understanding how these vectors work, the immunogenicity, the repeated administrations, the safety, the production to gene standard. That's why gene therapy received a hit in the 90s. But I still understand because we faced that when we come up with these new particles, because we, instead of rushing to the clinic, we decided to improve them, to get them ready for clinical trials. But the other limitation is how to get funding. People can give you funding for something that can go to the clinic, preclinical studies, safety studies, but it's difficult to get funding for vector development. So now we have these particles, another challenge, how to produce them to GMP standard, because it's very expensive. We need millions. Then if you go through the academic track, the whole process takes time. because you have to apply for funding, and that takes time. There is the other track going through spin outs, which is faster. But I can tell you that will really spend a lot of time just to get there, because to get all this funding, the whole thing takes time to cover the patents, to get the approval from the regulator. It used to take 6 months, now it takes 1 year. And to get the production to GMP standard, for instance, now it has been approved in the UK and Europe to produce, for instance, phages at GMP standard, but it took many years to get there. So I believe the fast track would be through spin outs and startup companies to get faster to the clinic. So if I look back, I think I wouldn't go through the academic track. I would go through the industry track to avoid all these years trying to publish these big papers in order to attract funding, or these high impact journals, which actually I don't know how they measure the impact, actually. And they can say that, like, PCR was never sent out for review by Nature. It was not sent out by Nature, and you can see the impact of PCR. So that's the whole process of time, how to get there. It's one after the other. Yeah.
Thank you so much for these insights. And the interconnectivity between like vector systems and cell therapies, I expect has also put a lot of attention to vector capabilities and should foster attention going forward. I would like to open the panel to additional questions from the panelists. If there are no questions, I would like to close the session, and I would like to thank the organizers of this excellent Medical Biotech Summit for enabling this interesting session, and the speakers for their presentations and valuable insights. We have heard quite a bit about the experience with CAR T-cell therapies, which is one of the early entrants of cell and gene therapies into the clinical practice. And we have also heard about the opportunities of scientific developments and innovative technologies in the field, including pluripotent, induced pluripotent stem cells and phage methodologies. While the administration of an investigational gene therapy to a patient had happened more than 30 years ago, the field is now in a rapid and dynamic phase of scientific revolution and evolution, with continuous efforts to pioneer. The development requires close partnerships between many stakeholders globally to bring this innovation to patients, and there are unique needs and opportunities linked to these fields, which are also aligned with the Saudi Vision 2030. And with this, I would like to close the session. Thank you so much.
Thank you.
Thank you, Dr. Mariam, and thank you all for sharing with us your insights and views, focusing on the improved understanding of new advances in cell and gene therapies that offer the potential to transform medicine. Everything has been shared in this session is very promising, so thank you so much. And now, ladies and gentlemen, we're about to begin a 30-minute break. A quick reminder, you can follow the conversation of our sessions on Twitter at RGMBS2021, and also participate in the discussion by using the hashtag RGMBS2021. We'd love to hear from you, so share with us your feedback and views. We'll be back at 7:35 p.m. Riyadh time for the last session of the day, an exciting one, so stay connected. Thank you so much.
The pandemic has taught the world of the importance of science.
And our mission is to improve the health of our communities,
including a clear message about how the benefits will flow to the community more broadly.
Riyadh Global Summit for Medical Biotechnology aims to push the boundaries towards a targeted and sustainable medical biotechnology enterprise in the Kingdom of Saudi Arabia. Let's hear from the Summit Chairman and the Summit Vice Chairman.
Riyadh Global Medical Biotechnology Summit 2021 present truly a fascinating program as a blend of sessions dedicated to strategy development, business development, as well as other sessions dedicated to technology development, infrastructure development, to serve truly establishment of medical biotechnology ecosystem vibrant in the country, as well as other sessions dedicated to scientific topics such as genomics, genetics, gene editing, stem cells, and others. All of those are being delivered by top leaders in the field from global pharmaceutical company and biotechnology companies from around the globe, as well as leading academic institutions with the aim to forge global partnerships that are of interest and will impact the country and the rest of the world.